What's up, AP Bio Penguins? So, this is the Unit 1 Q&A. Sorry. Hi, all. This is the Unit 1 Q&A. And in a moment, the APsolute RecAP is going to be joining us for our Q&A. So welcome. I'm so happy you all have joined us. So go ahead, and as we wait for the APsolute RecAP to make their way in, go ahead and tell us in the bottom in the chat, where are you from? Where are you from? That's your you're not going to be in my live. I'm sorry, sweetheart. The APsolute RecAP is going to be joining me tonight. So tell me where you're from. And if you have any questions, be sure to go ahead and go live. APsolute RecAP. APsolute RecAP, come on, join us. So if you have any questions, put them in the little question box down at the bottom. Ooh, I see we have somebody from Canada. That's so exciting. I'm so happy for you to join us. Hi, APsolute RecAP.
Hi, Tiffany. How are you?
How are you doing this evening?
I'm good.
I'm wonderful.
This is like deja vu, right? Doesn't it feel like 2020?
It does feel like 2020. It's crazy that it was almost, a month from now that we all went into quarantine.
I know.
And both of us started our big review stuff, right?
Which we both started before COVID. Ironically, it's like we had some sort of premonition, and then all of a sudden, the world shut down and everyone needed things.
Help. Yes. So I see the Marco Learning has said hi. So hi, Marco Learning.
Hello.
Of course, we love Marco Learning. So if you are needing resources for SAT, ACT, go check out Marco Learning. And before we get too far in, Melanie, will you tell us a little bit about the APsolute RecAP?
Sure. So welcome, everybody. Hello. Thank you for coming to the first of what will be several lives. And on Super Bowl Sunday, I think we have like 2 minutes till halftime, so I expect a few more of our friends to join us then. So my name is Melanie Kingett. I am the co-founder, so you can see my shirt, of the APsolute RecAP, which is a podcast and review company. We started with biology last year, so if anyone here is a listener of biology, just give us a wave in the chat so I know. Thank you for listening. And so we have really short 10-minute clips on a variety of topics for biology. We also have chemistry, government, music theory, and physics 1, all of which you can find on like any podcast platform like Spotify, Apple Podcasts, and Audible. And we will be having review classes in April to get you guys ready for the big exam. So you can check those out on our website.
And that podcast was the APsolute RecAP. And you can hear that on any streaming platform.
If you click on our name and Instagram, it takes you right to it. So it's a pun, right? So the APsolute
Internet
AP.
issues again, Melanie.
Listen, biology lends itself to puns.
So in case you weren't with us last year, Melanie has internet issues from time to time. So just bear with us if we lose her. But so tonight's thing is on Unit 1. So we figure we do a big Q&A. Any questions that you have over Unit 1. I know that if you're following along on my Instagram that you have finished Unit 1 with all of the review. So if you have any questions on Unit 1, go ahead and pop them down in the chat. Before we get started, I did want to go over the exam, kind of the general format of it, so that we are all kind of in the know. So I'm going to flip my camera real fast for everybody, and I hope that it shows up for you. So the exam is going to have 2 parts. The first part is multiple choice and is 90 minutes, and there's 60 questions, and that's going to be worth 50% of your exam. And the second part is going to be free response. And again, it's going to be 90 minutes, 6 questions, 2 long and 4 short, and it will also be worth 50% of your exam. The long ones are going to be 8 to 10 points, while the short ones will all be worth 4 points. Okay, so there is a little bit to know about those. And if you go and look at the AP Biology CED, so search on Google.
Hold on, wait, here we go.
Oh, that beautiful binder that they sent some teachers. And I have mine nearby. Oh, wow, where's mine?
I forget that I own it 9 times out of 10. And the best part about downloading the PDF is you can just do Control F and you can look for a keyword. And it tells you what page, what unit, all those things. It's a really great way to find connections between concepts because it'll show you, oh, this word is mentioned, 76 times in the CED. That means something.
It means it's important. But don't get too tied up on what's actually in the CD, because sometimes there's stuff that is underwritten. Like, they don't specifically say a certain word, but it's actually in the content. But so if you go to the CED, it'll tell you how those free responses are going to all be laid out. Okay. So also another thing that they tell you in the CED is they tell you how much the weighting is going to be for each of your units. Okay. And so according to the exam weighting, there's percents are up there. I'm not going to read these to you, but the percents are up there for you. Okay. And what I did is I went through the 3 exams that we as teachers have, and I have found out exactly how many the average was of those 3 from those 3 exams. And so you can kind of see that unit 1 has about 6, unit 2 has about 7, unit 3 has about 9, and then unit 4 has about 7. Okay. And the same thing is if you go to unit 5, 6, 7, and 8, you can see about how many their average was. So 6, 8, 9, and 8. Okay.
So Tiffany, how do you use that information to inform your teaching?
So I'm using that to kind of help my students to say, okay, you really need to know about cell energetics and you really need to know about evolution. But it also helps me to know that, okay, the test is pretty equally weighted between all the units. So they're all, to me, pretty much important. How are you using that information?
It's always a favorite question. Is this important? And will this be on the test, right? The 2 big ones.
Yeah.
And as teachers, we always say, well, it's all important. However, no, I use it more for pacing, but also you have to bring back unit 1 almost always. You know, you can never move on from a unit. It has to be incorporated in whatever you're studying. The questions are not going to be singular. They're going to extract from multiple concepts almost always. So.
Okay. So do you want to head into these questions and start kind of getting some of these answers for the students?
Sure.
Okay,
that sounds good. And for those of us who are here, Tiffany has questions that were submitted ahead of time, but if you would like to add to these questions, there's a question box right on the bottom. Please do put the questions there instead of into the feed, because we'll lose them there. We want to make sure we get to them. It's going to cover up my face too, so I'll goodbye when the questions
Yes,
pop
it will.
I'll try to.
I've discovered it actually doesn't cover up your face. But I have discovered that when I export this live and I post it online, those questions are not going to be in the video. So I'm going to read the question before I answer the question so that everyone knows what that question is in case they're listening to this later. So what makes up the macromolecules, and is there any easy way to remember those? Whew, that's
Do you have a mnemonic?
Question. Not really.
No.
Do you have any mnemonics?
I have a mnemonic just for the elements, right? So we're referencing the elements that are in them. You know, I use schnapps. I've heard people use schnapps or something like that. Do you similar?
I
just for
I use schnapps.
Schnapps, yeah, which is, you know, a fake word, but it reminds you of your main elements.
Yes,
kind
but
of
it stands for carbon,
hydrogen,
oxygen, nitrogen, phosphorus, and sulfur. So schnapps really kind of works to remember that and to learn it.
It does, and it also tells you kind of like relative proportion, right? So obviously these are carbon-based molecules, so carbon should show up a lot.
And I believe in every single, all 4 of them, you will have carbon, hydrogen, and oxygen. Okay. Yes. The important thing to know is that nitrogen is going to be found in both the proteins and the nucleic acids. Okay, so you need to make sure that you know that nitrogen is in those 2. So again, nitrogen is in the protein, and it's in the nucleic acids.
So does anyone in the chat know why? Let's see if we can get the answer to that.
Where is the nitrogen?
Where is the nitrogen in proteins and nucleic acids? See if we can't get that answer somewhere in the comments.
And I know that there's a delay between when we say something and when they start typing. So I'm going to go ahead and also say some of the other parts. So for example, phosphorus is only going to be found in the nucleic acids. And then it will be found in one of your lipids, your phospholipids. And of course, phospholipid tells you it has a phosphate. And also we see that the sulfur is only going to be found in your proteins. So I have seen questions where they will give you the information about the different elements, and they want to know which macromolecule is this. If you see a sulfur, you immediately know that that's your protein. If you see that there's a phosphorus, you pretty much know that that's going to be, nitrogen and phosphorus, you know that's going to be nucleic acid. So great job. I do see there's a lot of questions coming up in the chat. I want to remind you that if you're posting a question, put it in that little question box, because I will tell you, I only see pretty much one person at a time before they scroll up. because of
And
the double screen.
I'm going to start, I was just in the chat. We did get correct answers for the nitrogen, so amino group, and the nitrogenous base. Great job. And I think that's one of the lovely things about science in particular is we're just not super creative with the way things are named. And that's lovely, because it's either going to be named after what it does, what it looks like, or a person, you know, when it comes down to it. So,
and then, so carbohydrates, the way I remember that is it's made up of carbon, and then a hydrate, and hydrogen is a hydrogen and oxygen. So it basically tells you what it makes up, as you just said a moment ago. So great job there. So I'm assuming there's gonna be more questions about specific macromolecules. So we can come back to that, because that's such a loaded question. So let's see. So let's think about the proteins for a second.
So this question.
I'm sorry. No, it actually came through your account.
I know.
And then
I know.
you told me about it. I was like, let me go ahead and pop it in there so we have that.
I wonder if it was one of my students. We have claims that they want to memorize all of them.
That's a great idea, Miss Pratt. Miss Pratt has written in the chat. She wrote nucleic acids and proteins are the only two macromolecules with the letter N. So there's going to be no N in carbon, and then there's no N in lipids. That's a great way to remember that nitrogen is only going to be found in your proteins and your nucleic acids. So great job. Thank you, Miss Pratt. I appreciate you. You're so awesome.
Never thought of it that way.
No, she's great.
That's fantastic.
Okay, so speaking of proteins for a second, the question that came up is, do I need to memorize all the R groups? So first thing, what makes up a protein? So protein is going to have amino acids that's its monomer, okay? And so the monomers, the amino acid is going to have the amine group, a hydrogen, our carboxyl group, and an R group. That R group is variable. It can change. So Melanie, do they need to know all of them?
They don't need to know what they are specifically, no, but they need to know how they behave. And so do not memorize them, because if you, I mean, a lot of students who take AP classes do so because they were really strong in prior groups, and they're likely really strong in prior sciences, sometimes because they're really good memorizers. And I think that's a wonderful tool. But understanding is where it's at. So no, please don't memorize the R groups. Would you concur with that?
I would have concurred with that. Please do not memorize them. I will tell you, even with my degree in biology, I never had to memorize the R groups. Now, I will tell you that looking at the side chains, I had to know, would they be polar, or would they be non-polar, or would they be charged? And that's the important part. And so if you see that there is an oxygen in it or a nitrogen in it, those electronegative atoms, you know that it's going to be polar. If you see a whole bunch of hydrogens and carbons and rings to it, you know that that's going to be non-polar. And the important thing is that polar was going to fold outwards toward the aqueous environment, versus non-polar is going to fold inward to kind of hide from the aqueous environment. And then, of course, charge is similar to polar, because if it's charged, it's, of course, polar.
And that's going to be really important.
I lost her.
Oh, can you hear me? Am I here?
I got you now. We're good.
Okay. That's going to be really important for later units when we talk about point mutations, or we talk about, you know, amino acids being modified or changed. You end up with a different chemical property. Structure influences function. So we have to draw that thread to the final product.
Usually it's not going to have equal amounts of carbon and hydrogen, because of the fact that carbon can make 4 bonds and hydrogen can only make 1. And so usually if you're looking at carbon bonded to other carbons, it'll have a maximum of 2 hydrogens. That's why if you look at the structure formula for carbohydrates, it's CH2O, and is always in that multiples of 1 to 2 to 1, because the carbon has to have 2 hydrogens on it.
And
I'm sorry,
we
I just
mentioned,
caught that message.
No, we mentioned the CED earlier, and I do want to point out one of the things that it'll have is exclusion. So things that you don't need to go, you know, don't wander down the path in that direction because it's unnecessary. So one of them being the actual chemical structures, which answers this question. So you do not need to know the actual chemical structures of the R groups, but you do need to recognize their differences, would be the differentiation there. Hi, everyone. So if you're just joining us, I'm Melanie from the APsolute RecAP. We got Tiffany from AP Bio Penguins, and I think it's halftime. So
ooh, halftime. This is a halftime show, right?
This is
We are the halftime show.
We're the halftime show.
I'm sorry, guys. It's a little late, and we've all been virtual teaching for way too long in our lives.
Yeah.
Anyway, so the next question we have is, what are disulfide bonds? So I wanted to go ahead and throw this one out there since we were already on the conversation of proteins. Disulfide bonds are literally just bonds between two sulfates.
Right? Di for two?
Yeah. And it's part of the tertiary structure in proteins. So proteins have four structures. You want to go over them for us, Melanie?
Sure. So we got primary structure, which is going to be your chain of amino acids. That's going to be formed during translation at a ribosome.
What kind of bond?
A peptide covalent.
Ooh, okay.
Then you have your secondary structure, which is going to be your alpha helix or beta sheet, and that's due to local R group interactions as compared to, what, did I say something incorrect?
Yeah, the secondary is your hydrogen bonds.
Oh, goodness. There you go. See, this is why there's two of us.
Yes.
Why don't you take it from there?
It's okay. I'm sure you have it.
Then you have your tertiary structure, which is going to be your folding. And then your quaternary has more than two different chains interacting together. But all of those pieces relate back to the R groups at the end of the day, because that's going to be your variable within one amino acid to the next.
So there's a partner teacher of mine who used to play Stump the Chump. And her big thing was she wanted to say wrong things all the time. So Melanie was honestly just testing me to make sure that I was going to catch her. So Melanie knows this. Great job, Melanie. Thank you for trying to stump me. It's okay. I was paying attention.
You're welcome.
I got you. So disulfide would be in our tertiary structure. And again, if you have questions, throw them down into that box.
It's still local. It's still local folding, though, which is high.
It is still local folding. Yeah. You mentioned the R group, and the R group is where we had the issue. There's a lot of questions about bonds in this. About proteins in here. Whew.
Well, while we have the mention of bonds, can we just give a nice love moment to chemistry teachers and our hydrogen bond phrasing?
Hydrogen bond phrasing? What?
It's not a bond.
Oh no, yeah.
Land. Yeah.
It's actually an attractive bond, just like ionic. Ionic is not a bond, it's attraction.
Yeah.
It's a positive charge and a negative charge, and they're attracted to each other. Sorry, I was going through the questions and I got distracted.
Intermolecular force. It's funny because since one of the podcasts I'm recording is chemistry, and I'm not writing chemistry, I've been getting quite the brush up on chemistry, and there was a whole paragraph about hydrogen bonds and biology versus chemistry, the way we phrase things. It's like our starfish, right? It's our starfish.
Okay, so while I'm going through the next question, the question that came up is, what unit are most schools on right now? So I know that every teacher teaches in a different order. Okay, so as everyone is typing in the chat what unit they currently are in, keep in mind that some teachers have started with evolution or ecology, while some teachers kind of jump around, or I kind of condensed them all. So 4, 5, and 6 was all for unit 4 for me. So when people are saying down the chat what they're in, but Melanie, what are you currently? What unit are you in?
Yeah, so we're testing on unit 6 this week, and I am going in order. But schools also started very different times. We didn't start until the very last days of August, whereas what was your first day of school?
The very last week of August.
Oh, right, you were later.
We got pushed back. I got pushed back an entire month in my start date because of COVID. So and we're actually we're gonna finish unit 7 this week. So later on this week, I'll actually have my unit 7 live stream to go over unit 7.
So will 8 be all you have left, or did you do 8 already?
8 will be all I have left, and then I will do a little hodgepodge unit to try to connect it to the animal systems, and my goal will be to finish before spring break. So my students will just have review from spring break until the exam, and I'm going to tell my students to take the first test, so that May 14th test is when I'm going to tell my students to take it. So I'm seeing a lot of units. Y'all are good.
Yeah.
You can talk, Melanie.
Yeah, for those of us who haven't heard, the College Board did announce more testing dates for their exams. Bio got 2 more test dates, but they are digital options, not paper options. And whether a test got paper or digital as those second and third options really just vary. If you are a very math heavy exam, that's going to be like your chemistry, your physics, or your maths, calculus, statistics. They are going to really have more paper than digital. But the digital test leaves a bad taste in my mouth with the way it's designed this year. You know, any teacher will tell you, go back and check your work, you know, in some capacity, you know, but you can't do that in the digital test. You have to move forward, and you cannot go back. And that's just like one of the reasons. You also will not be graphing, which you're missing a whole, they're not going to have graphs production on the biodigital test this year.
Oh, I missed that information. That's all of what question number 2 is.
Yes. So they instead are going to put that skill in other places. So you're not going to be producing any graphs because you cannot handwrite any answers this year. If you took an AP test last spring, that was one of the choices. Everything has to be typed. And so that is going to be a hindrance. So take the paper test if you can. I think
I recommend paper.
I would recommend it.
So real quick, while we're talking about the units, Unit 7 is evolution and Unit 8 is ecology. So I'll flip the camera back because it's still on my screen. So in case you were wondering what the units were, Unit 1 was chemistry, like biochem, which is what we're talking about tonight. Unit 2 is cell structure. Unit 3 is cell energetics. Unit 4 is cell communication and cell cycle. 5 is heredity, 6 is gene expression, 7 is natural selection, and then 8 is ecology. Sorry.
And your teacher might not be saying any of those names to you, and that's okay. It doesn't matter. They might be using chapter titles. You might be dividing it up very differently, and it's all tomato.
Yeah, as long as you're learning the content, it honestly does not matter what unit you're in. Okay, so let's get back into some biology. Well, actually, this is actually chemistry because we're in the biochem unit, right?
Carboxyl.
So what is a carboxyl group and why is it important? So
you can take that one.
I can take that one. Great, thanks. So carboxyl group is a carbonyl plus a hydroxyl. So that's why they make it a carboxyl, because it's a carbonyl and a carboxyl. So the carbonyl is the carbon double bonded to an oxygen. So it's kind of like you are a carbon and your 2 arms are going out to a steering wheel, which is your oxygen. So carboxyl, carbon, okay? And then the hydroxyl is an OH group. And so the carboxyl is a C, double bond O, OH. So my partner teacher used to always talk about that you are driving your car, eating a hydroxyl cookie. It never worked for my brain, but her students absolutely loved it. So
like the Oreo, like the knockoff Oreo Hydrox? Got it.
Yes. So she tries to and that's what all the students love. So I always try to throw her little reminders in there because she was so great at that. So why is it important? Because it's polar. So the big thing about it is that it's polar, which is then going to cause all of those groups to be polar also. So whenever you're thinking about making bonds, so we mentioned earlier that the amino acids were going to be paired together with a peptide bond to make that primary structure, to make the proteins, of course. So the OH group is going to be taken off of the carboxyl, and then the hydrogen will be taken off of the amine group of the next amino acid.
If you think you know the name of that process, put it in the chat. If you think you know the name of that process, put it in the chat. Removing.
Oh, yeah. Great job. Because that's also in this unit. Knowing what that process is called, the creation of water when making a bond.
Yes.
Ooh, what is that called?
It has a couple names.
Oh, yeah, you're right. There are a couple names. Well, we're waiting for them to type in those. Someone says, can we use scratch paper on the exam? You've read a little bit more than I have about the exam. Can they use scratch paper on the online exam?
Yeah, I don't see how you could be stopped, but it will not be included in any. Good job, guys. You're all getting them right. Dehydration, synthesis, hydrolysis. Let's go back to the word hydrolysis in a second, because that's also important to this unit. But scratch paper, yes, you can use scratch paper the same way that you can still use a calculator for digital.
So dehydration is when the water is coming out. So if you get dehydrated, it's because you're losing your water. Same thing is it's a condensation reaction, because when you're thinking about can of Coke condensing on the table, the water's coming out of the air. I don't know why I put hyphens over that. Versus hydrolysis is like the hydrogen, it gets like
Yeah,
hydrolysis.
karate chop, lysis,
Karate chop.
lysis to snip or to cut. So to cut up with water, breaking the bond and inserting what you have in H2O onto either side. So you can
hydroxyl
see
for
that.
one and hydrogen for the other. And those paper tests will be in person. There's no way to do a paper test virtually.
Nope.
Because I'm assuming with the new platform they're using for the virtual exam, even though it's not the same platform as last year, because you don't need internet for it, right?
I have to read more on that. I have heard you don't need continuous internet, but it's an application that they are going to require for you to download and install onto your device. So
I'm assuming the timing built into that.
I think you have to do it ahead of time.
No, I meant the timing on the exam.
Oh, yeah.
Like a paper test at home, they couldn't time you. No. Nor could they keep the security of it.
No, and speaking of security, there's a lot of things they're going to be doing. The camera being the first thing that's different this year. You will have to have an ID, and this is true whether you take it at school digitally or at home digitally. It's going to have the same requirements. Any kind of
okay. So back to some biology. What bonds do starch and cellulose have, and which can be broken down by animals? Whew.
So you find the differentiation between this important for the exam?
I do.
Why?
And I'll get into that in a second. So the starch and cellulose, number one, they're both in plants. So that's easy peasy, okay? Starch is going to have an alpha bond, while cellulose is going to have a beta bond. Now, knowing what kind of bond they have, not important. Okay, you don't have to be able to say, oh, starch is alpha and cellulose is beta. And it's really honestly not important to know that that's called a glycosidic linkage. Now, the thing that I find important though is knowing about why, and you can't break it, but how are animals that do eat wood or plant material, how are they able to survive? And the symbiosis and the ecology of it is where I find the importance of it. Okay, so we can't break beta linkages. So you can't break down cellulose, and no animal can break down cellulose. But animals like termites, they eat wood. So logically, how do they do that? And so there's a protozoa, I believe it's a protozoa, that lives in their intestines. And so when they eat that wood, the protozoa breaks down that wood and releases out the glucose molecules. Because all cellulose is a big, long chain of glucose molecules. I believe it's actually branched. I can't remember, but I think it's branched.
Cellulose is branched, yeah.
Yeah. And so it breaks down those glucose molecules and releases out all of the glucose. And so then the termite can absorb the glucose and the protozoa got food. So it's a good example of symbiosis. And so that's where I find they may have a connection. Yeah. And they say, oh, termites have this organism that lives in their intestines. It does this. Why does this happen? And you would have to make the connection. Okay, well, termite can't break it down. That's why they're doing it and go through the whole process. That's why I think it's important. Yeah. But the actual knowing alpha versus beta, not important.
And I think that's a great example of how structure influences function and relates back to the bigger ideas. And I think similar to how we talked about proteins, it's not necessarily about identifying the specific amino acid R group, matching them up. However, understanding the bigger picture is where the emphasis needs to be for the exam. Okay.
So yeah, somebody said about glycosidic, phosphodiester, and peptide. Of those three bonds, I would definitely know peptide bond.
Yeah.
But the other two, I don't really find them to be that big important to know. If you really need to make sure that you remembered it, glycosidic makes you think of glycogen and glucose and other sugars. Phosphodiester, hello, we're talking about phosphate. We just talked about nucleic acids have phosphate. That's the only one that does. So, there we go. And then of course, peptide.
You might see these terms in larger questions, and I think that's kind of like the where the vocabulary can go sometimes. It'll be kind of like vocab overload in some of the questions. Your role as a student is to get through the weeds and find the ones that give you the meaning of the question. So enzymes, organisms, things you've never heard of before, even processes you might not even be aware of. But the College Board would not be asking you this question if you didn't have the foundation to figure it out. And knowing that, kind of going into it with that mindset is going to help you get to the end of the line, and not getting bogged down in the preamble of the question. As it goes.
They will definitely do that to you. And while we're speaking about the preamble and the long prompt, okay, when you see a long prompt, just take a deep breath. Okay, remember that you know this stuff, and they're looking at your biology knowledge. Okay, so a lot of the stuff they're going to tell you and explain a whole bunch of stuff to you. Just underline, circle it. You can do this.
Another reason to take the paper test.
Yes. Online, you
Annotate
can't do anything to it, right?
Yeah.
I definitely agree. Paper is definitely going to be like why I tell my students to do it. But, you know, depending on if you've had a lot of COVID out, you know, if you've been out of school for a lot for COVID and you've had trouble keeping up with things, it's completely up to your school how you do it. I believe that the schools are deciding, not the students.
It is the school, but it can be different per student. So like all students who take AP biology at your school do not have to have the same test date. It can be distinguished for different students.
Right. So let's talk about water for a little bit then. I see there's a question in the chat, and I'm not going to do that one. I'll go back to, I promise. How long are we going to be live for? We'll probably be live until about 9.
I don't even know what time it is. So
it is 8:31.
Okay.
And we'll be like I can't, like, my screen did something weird, but I can see the time still. So we were planning on staying about until 9, unless you run out of questions, and then we'll just like bye. But I don't think we'll run out of questions. So thank you for saying we're amazing. We do try our best. No. Anyway, so the question is what is the difference between adhesion and cohesion? Hi, Ms. Kaufman.
I'm a nerd. I majored in Latin in college. That was my side hustle next to bio. You're a nerd as well. And what were your majors in college? Listen.
Biology and chemistry. I'm a dual degree.
Yeah. So when I see the words adhesion and cohesion, I just go right to the prefix ad and co. So ad means to, and co implies with. So to adhere is to adhere to something else. To have cohesion is to stick. I know I said stick. You don't like when I say stick. It's to come in. It's
you know that about me. That's surprising. Wow.
I think you've yelled at me before. To have cohesion is when water molecules attract to each other through their hydrogen bondings. So why does it? So why do water molecules adhere to things that are not water? That's a good question.
Where'd you see that question at?
I didn't. It's my teacher brain going to the next question.
Yeah, you're good. Well, so like when we think about adhesion, it's going to be adhering to other things that are polar. And so if something is polar, then it can adhere. So thinking about a graduated cylinder, it's a great place to see it. So when you pour the water in there, the water doesn't stay exactly flat, right? You have to go get down to eye level because it does this stupid meniscus thing and it climbs the walls. Why? Because the silicon dioxide that makes up your glass is polar, so the water is attracted to it. Similar to when we think about capillary action and how the water makes its way up to the leaves from the roots. So it has to do with the cohesion that each water molecule is attached to another water molecule. And then the sides are basically also attached to it. So it can kind of climb the wall, and then also it pulls from the negative pressure in the leaves.
Transpiration off the top, little different unit. We'll talk about osmolarity.
Yes.
I always think of it like barrel of monkeys a little bit. Like they're almost like pulling up on each other. I loved that toy as a kid. That's a good one. Yeah.
It's a good one. We got some more that just popped into the thing, but I wish they would go in order. Like I never can find them. Like I want them to be in order. Okay, so let's do this one for a little bit. What types of graphs, tables, diagrams can be associated with this unit? Well, okay, a table that you could have maybe is that there was a scientist who investigated this sample, and it had, you know, approximately this materials in it. Maybe if they say, you know, we've got, I don't know, 25% carbon, 50% hydrogen, 25% oxygen, you can say, oh, CHO, carbohydrate. Or if you see that they say something about, they talk about that there is, I don't know, 30% thymine, 30% adenine. I've lost my math, but 20% cytosine and 20% guanine. Okay. Yeah. And you can say, oh, that must be DNA because you have equal amounts of your complementary base pairings. First, if they had different numbers of A, T, C, and G, you go, oh, that must be RNA because of the fact that they don't have the equal pairing. So that's something you could have. Enzymes aren't actually in this one. I just teach them in this one. Sorry.
Yeah. Oh, you know what the other one? So, because you mentioned DNA, I do want to talk just super briefly about like the history of DNA. Not explicitly here, but they could talk about, Mendelssohn experiment, or they could talk about like these historical ways in which they came to find DNA. So, Griffiths, Avery, all of those scientific experiments to deduce between proteins and nucleic acids as to which was the molecule of heredity. So kind of like reviewing some of those wouldn't be a bad idea, just to understand it yourself as to how and why the results made sense, because they could use something like that as well.
Somebody in the chat said chi-square. Well, chi-square is valuable in any of our units.
Yeah.
You should definitely be aware of how to do chi-square, because if I remember correctly, isn't it one of our science practices?
Yes.
I think like literally chi-square is a science practice. So it would not surprise me if there was a chi-square question on any of your units. Usually they're going to be like, teachers usually teach them in the genetic stuff. So they would want to switch it up and not do it in genetics. So definitely chi-square is
somewhere
valuable
unfamiliar.
tool. Yeah.
This
unit's really hard to pull up.
Yeah, I think that was to my point earlier, that this kind of overlaps with other places. You're going to kind of see it interwoven in later units.
This is the foundation.
Foundation. My chi-square. So I do have a podcast that's just like as a math review for all of the big mathy concepts in biology. I have no idea what number episode it was. I'm going to say 20 something. But we have 69 episodes out this week, so there's plenty to choose from.
Gotcha. Okay. So I see there's one in the chat. I don't want to lose it. It says, are lipids majority hydrogen? They're carbons and hydrogens. So I wouldn't say they're the majority hydrogen, because remember, hydrogen can only make one bond. So I would say carbons and hydrogen.
Lipids have a lot of diversity in what is categorized as the lipid. So in terms of what they have in common, being non-polar primarily. When we get to phospholipids, you have amphipathic chemistry here.
Oh, it's actually one of my babies. This is one of my real penguins. Like, you are all real penguins, but this is one of the in my classroom penguins. So will we need to directly reference scientists' names and FRQs, or just the experiment? If I remember correctly, the actual scientists' names aren't in the CED, nor is the actual experimentation. But what the writers are going to do is they're going to pull a lot of that historical information, and they'll describe to you the experiment. They'll describe to you what Meston and Stahl did. They'll describe to you what Hershey and Chase did. And from that, you'll have to make connections to the biology. So don't think you're going to walk into a question and say, well, the Hershey and Chase experiment, what was proved? I don't know. So you don't actually have to know.
Who killed the mice? Which one was the mice and which one was
Griffith was the mouse.
Oh, I wasn't asking you.
And he injected them, and yes.
History of science has a very bloody past. So to the point, no, like Tiffany said. However, always reference back to the information in the FRQ, right? You don't want to just like pull things out of thin air for your knowledge in some capacity.
Also, speaking about that, sometimes they do experiments that you may have done in your classroom.
Yes.
Okay, so don't try to pull off of your own knowledge. So just because XYZ happened in your experiment in your classroom, maybe that wasn't what was supposed to happen, number one. Or number two, maybe they were looking for something else, so they've given you different data. So you definitely want to look at the prompt that they've given you and pull the information from the experiment that is on the test, not from your experience. Your experience will help to better understand the question, but don't rely only on your experience from these experiments.
One of my favorite times, I was grading something on evolution, but it was essentially about vestigial structures and the whale hip and, all that. And the student went on a total tangent, started telling me about all these things they saw on Animal Planet. And they were all correct things, but as part of their evidence, they're like, no, the evidence from the question, not just evidence, which is pretty great.
So someone has asked, can you go over nucleic acids and their structures? So before you get into that, let's talk about the main things that make up a nucleotide. So what makes up a nucleotide now?
So you're going to have three main parts. You're going to have your pentose sugar, your phosphate, and your nitrogenous base. The pentose can vary. You might have ribose or deoxyribose. You're going to have differing bases as well, depending upon whether you have DNA or RNA. And what the correlation here, using the word nucleotide, is that it's the monomer, right? Monomer of a nucleic acid, which is your polymer. And one thing that people, not people, tend to forget is, where does ATP fit into all of this world? And what is ATP made of? So obviously I brought it up now. So ATP is going to fall into this realm because of its structure, but it's not going to form a polymer. Oh goodness, what sugar is it?
If you don't know, I do know, don't worry.
I don't think I know.
It's a ribose.
All right.
Yeah, it's a question I asked for my Magna Bio kids. My regular level bio kids had to know it. So.
Oh my goodness, you're rough.
You're
have to know tough.
I am not you, but I mean like the students need to know it. So with it, it's going to be the ribose, and then you'll have three phosphates on it with ATP, because it's adenosine. Because it's that when you think about adenosine, it's just the sugar plus your nitrogenous base, which would be adenine. And so also, think you mentioned ATP. Do you know there are other things? There's GTP, and there's TTP and CTP. There's all of those, but what they did, they being the authors of these books, is they just made them all ATP, even if it's one of the other ones, just to make things easier for brains. So it's not always actually ATP, but we do always use ATP in our examples for the most part.
Functionally, follow the path. It's the same.
So
same concept.
Big things about DNA versus RNA. So you mentioned the nitrogenous bases. So with RNA, the nitrogenous base is different, is it's going to have a uracil versus in DNA, it will have a thymine. So when you're looking at multiple choice questions, and it's talking about which one of these is the RNA, or which one of these is your coding strand, or which one of these is whatever, okay? And you're looking for something about DNA. If there's any U's in any of those, you can automatically knock those out. Those are not possible answers. Same thing as the reverse is true if you're looking for something about RNA. If there's any T's in them, those ones aren't true, and you can just go ahead and knock them out. Okay. Another reason why I take a paper test, because you can't knock the answer choices out online.
Yeah, that's true. Yes. Oh my goodness. I wonder if there'll be any. I mean, we're going to find out more in a couple weeks, but I wonder if there will be like any sort of interactive features where you could do that. That would be helpful. Like, it would be at least getting rid of choices.
Okay. And I believe there's a Katie that wrote something in the chat earlier wanting to know about purines versus pyrimidines.
Well, you have a very strong opinion about Georgia.
Okay, I've changed that for you, okay? Good, I've changed it. Anyways, there's something, I cannot remember which follower said it, but one of the followers said that the pyrimidines have Y's in them. Thymine and cytosine, which are both pyrimidines, have Y's in them. That was great. And I said, well, what about when you're talking about RNA, the uracil? And they go, oh, well, T and U substitute for each other. So if you know T, you know U. I'm like, okay, fine. But the thing about it is that purine is a small name, but it's a big structure. It's going to be the one that has the double ring, versus pyrimidine has a big name, but a small structure, only has the single ring. So I always remember that those are kind of opposite of each other.
Yeah, I always try to think of... Go. Yeah, go ahead. Go ahead, Georgia. Tell us all about it.
Okay, well, the Puritans went to Georgia. So purine and G and A, and we're not going to say the other one I came up with because you didn't like that one. So whatever. And then the pyramids are cute, by the way. Pyramids are cute. So C-U-T and then a little E. They're cute. Yeah. So remember this. So
I always also try to have a mnemonic that correlates to double and single, right? So my ninth grade students take ancient history at our school, and so they came up with tombs and crypts are in the pyramids, and they have 1 point, and so they thought of that as one. And when that's just what was on their brain. I think they were being tested on something Egyptian at the time. We always go with gold is always pure, and we think of like wedding bands, like gold bands, as the double ring, as the association for us. But obviously gold It'd be
Oh, I
better if it was
see what
silver. Is
they are.
that's
They could be
why the
silver
gold
rings,
is all,
AG.
yeah. They could be silver rings. Maybe we switch it over. But they have to
Okay,
be silver
angels of
then.
God are
Depends.
pure. AG. Angels of God are pure.
All right.
Hey, you know what?
Experience. Very nice.
As you've noticed, there's more than one way to remember this stuff, okay? And so
And if
you as
you,
a student need to find what works
mhm.
for you and your brain. I can tell you until I'm blue in the face all the things that help me remember something, but unless it actually sticks and helps you to remember it, then it's not going to matter.
Not at all.
All right,
All
so
right.
let's see if there's any more fun questions in here. We've gotten a lot of questions done. We're rock stars.
Awesome.
Okay, so we haven't mentioned these words. So I do want to mention these words. Like, even though we've already talked about the R groups, I want to mention with these words, hydrophobic. Sorry, the question is, how do we determine if a group is hydrophobic, hydrophilic, or charged? So hydrophobic, if it's phobic, right, it's afraid of something. We go with your Latin words, right?
Yeah, I know.
So hydrophobic means that it's afraid of the water. So whatever is going to hide from the water, which means it's going to be non-polar. Hydrophilic, it's going to be polar because it's water loving. It loves the water. And then if it's charged, you're going to see there's a positive or a negative on it. It's
dead giveaway.
It's a dead giveaway.
So we didn't say where we were from, I think. So I'm from Pennsylvania currently. And so hydrophilic, I can't help, but Philadelphia is the city of brotherly love is our slogan. And so philos, yes, means love. And so that always comes up for our class. But Tiffany, where are you from?
I'm in Georgia. You couldn't figure that out. You know, Georgia. And I teach in Conyers, Georgia. So I tell my students, when they're trying to do their complementary-based pairing, ATCG at Conyers, Georgia.
Oh my
That's
goodness.
where I
it's
teach.
Like built in for you.
It's built in. And actually, I went to college at Georgia College. So it also worked there too, at Georgia College. I just keep DNA around me, and the world wraps around me too. Because when did you say that your school year started?
The end of August?
August.
Right?
And you know what the start codon is?
Oh gosh. Yeah.
Ooh, there you go. And I started in August too. So there you go. Like the world is wrapped around us.
That's true. I've never, it's funny because like the more you look for science, the more it finds you.
Oh yeah.
You can't.
So when you buy a new car, you see it everywhere.
It's I know that's true.
Okay.
Awesome. Cut the pie. Hold on. Not sure. Cut the pie. Oh, like for pyrimidine? Okay.
You cut the pie. Oh, that's a cool way to remember it. So thank you, Miss Unland. I think I'm saying that right. Oh, sorry if I didn't. Thank you for that way to remember. Cut the pie. I'm amazed. There's a lot of teachers following us. Like we saw Miss Pratt, we've seen Miss Kaufman, we've seen
my next question. Obviously, we have some obvious names out there that begin with Mr. and Mrs. But if you are a teacher or a student, can you just put teacher or student in the chat so we know who's here right now?
Yes, we're excited to have all of you, actually.
And hopefully, yeah, as we didn't make too many mistakes, and Tiffany's pointing out when I say something wrong.
Hey, I'm not perfect.
Listen, this is funny. I think what's funny. So last year, how do we even know each other? I guess we didn't talk about that. Instagram. We're Instagram friends. We both started doing things for bio at the same time, and I had asked Tiffany if she wanted to go live in the spring. To that point, I had only ever done live once, and we were kind of figuring it out as we went. And oh, great, I said it correctly. And it just kind of snowballed from there. We've been in touch ever since.
Yeah. Well, I've been doing my reviews with my students for 3 or 4 years. And then last year I shared my main account with the world and said, hey, come follow. And then 2 days into it, I was like, wait a minute, I should probably just put it on its own account. So last year I did a whole bunch of copying and pasting the entire review from one account to the other. This year, the whole thing's just on this account.
Yep.
So let's get back into some bio.
Sure.
What is the biggest difference between lipids and carbs?
By structure or function? Structure or function, I guess.
It just says, what is the biggest difference?
All right. So if we go back to the elemental components, you're going to find a lot of commonalities here. carbon, hydrogen chains oftentimes. And so I know that if you're looking at something like just like right off the bat chemically, if you see rings, you're like, is it a steroid? Is it a carbohydrate? You know, if you're looking at chains, you're like, is it a fatty acid or do I have something else? So there can be some differing components there in terms of structure, functionality. Yeah, go ahead.
The big thing when you look at their structures is that oxygen. Okay, so if you're looking at a big long chain of carbons and you see an oxygen in each one of those carbons, you know that you're looking at a carbohydrate. If you don't see a whole bunch of oxygen, you know you're looking at a lipid in terms of those long straight chains.
And it has to do with the ratio, the quantity you have as compared to the other.
Yeah. So I always tell my students with lipids is 1 to 2 to very few versus carbohydrates is 1 to 2 to 1. So
I like
that's an easy way to remember that one. And you were saying about the functionality of it.
Well, I mean, for carbohydrates, you're going to have energy storage oftentimes, you know, when we form our glucose, and then long-term energy storage. And you can have the same for some lipids. Steroids, the relevant thing for steroids too is that as a lipid and as nonpolar, they're going to pass through the membrane of cells when you're going to have other components that are going to need extracellular receptors and functioning as ligands. So were you answering the question in the chat?
Yes, I was not looking at you. I'm sorry. I saw the question and was like, let's go ahead and tell her no. Or tell them no. Because I was like, I don't want to have a student try to think they have to memorize. And
then your lipids, you're going to have your phospholipids forming your membranes. And so you see them being like key players in certain processes. Do you often find them intermixed? I mean, my students tend not to cross them very much.
Oh, my students mess it up all the time. They do.
Oh, okay.
They especially, well, they mess up naming, I guess, is the best way. Plus, they mess up the phosphates versus the sulfurs, and they mess all that kind of stuff up. But but you have to keep in mind that this is right when we learn at the very beginning. They always have trouble, and as we get kind of more into it, they get a little better. But biochem is definitely our hardest unit. Once you get to the end of it, it's our hardest one because of the fact
and I think some of that depends on did students take chemistry beforehand or not, and that's very different at lots of different schools. My
students don't take chemistry. I'm the first biology class that they take. They don't even have regular level biology. They come straight into AP. We just like, hey, here's the sharks. Let's go.
I mean, swim in deep waters, right?
So one thing, though, to keep in mind is that carbohydrates are polar versus lipids are non-polar. That's a big difference between these two because this is what's the biggest difference. Well, it's the polarity. That's like the biggest difference. You really want to get into it. The only thing that's even slightly polar in terms of a lipid is those phospholipids because those are amphipathic because they have that non-polar region and the polar region. And
that prefix amphi you guys should be familiar with, meaning both, right? So implying it's two sides.
It said we had a new question. Oh, okay. What determines if lipids are saturated or unsaturated? I'm taking pictures though.
No, I was bending my fatty acid.
Oh, okay.
Sorry, that was not overt. Yeah,
just taking a picture. I'm like, okay, let's take a picture of this one. Woo.
You just totally dated yourself because there are no cameras really anymore with like a shutter.
You're right. I did date myself.
So
I'm dating myself. I'm dating you too because are we the same age?
We are.
Okay. I'm sorry. So what determines lipids whether they're saturated or unsaturated? Okay. So saturated is completely saturated, completely covered in hydrogens. So every single carbon is going to have 2 hydrogens. Versus unsaturated is going to have double bonds in them. And so if there's at least 1 double bond in it, it's considered unsaturated. And as she was showing off her kinks,
my fatty acid.
That's what it was. You were doing one versus the other. I'm following now. Okay. And so you could have your kinks, which
carbon chain.
Thank you. Your double bonded carbon chain, which makes it unsaturated. And so things to remember about that is whether it's trans or cis, which I don't know if I've ever seen them bring it up, but a cis double bond is going to inhibit the compression more than a trans double bond. A trans double bond looks just like a normal straight chain, but the double bond is in there. I've never seen it come up, but they may explain that to you in a prompt and say, oh, you know, trans is going to, you know, have the normal bonding versus cis is going to have a kink. How does this impede the temperature of the membrane in which it freezes? And you would say, oh, well, you know, it's less likely to compress in the cis than it would in the trans. So
yeah, and that'll come back to fluidity, really, and that's the linkage to unit 2. Cell structure and
every
function.
time I try to like look at the chat and then it gets stuck on my screen. I can't get rid of it. I do that every time. Okay, so we got about 5 minutes left, so we're gonna. Oh wait, that's not what I meant to do. I meant to do a different one. Sorry.
I think that's unit 7 though has the most. And 3. Unit 7 and 3.
Yeah, 7 and 3 have the same amount. Yeah. Based on my analysis, also, actually, if you look at their math, 13 to 20% is unit 7, and then 12 to 18% is unit 3. But based on the calculation I did, they both had 9.3 questions. Anyways, so the question said, will the live be recorded for us to refer to back in another day? Well, of course it's going to be recorded. So
and
when we're done,
didn't crash.
No, because who hosted? No, I'm just kidding. So I will save this to my IGTV, whatever that little button is called. And then I'm also going to export it and pop it onto YouTube. So it will be in my channel. So if you go to my link tree, you'll find the link to get to my YouTube channel. I don't have a fancy little AP Bio Penguins on there, sorry. But if you search AP Bio Penguins on YouTube, I think you find me. And of course, I will give you APsolute Recap's little hashtag to handle thing. You can also follow her, because if you're not already following her, you should be following her to get your podcasts. Because I'm sorry, but even if you go for a walk, you get to walk your dog and you want to listen to some biology. That's how you can study. And the hard thing about studying sometimes is to get out of your bedroom, and especially you that are all virtual. Like my students have been virtual this whole time. So they literally sit in front of a computer all day long, and then they have to do their homework in front of their computer for even more time. Sitting, y'all just need to get up and move. And so with a podcast, you're able to put it in your ears and you can listen as you go for a walk and get some fresh air, get the blood moving. And it helps, of course, blood helps for your brain to remember and learn. So any last minute tips you got for them? We got 3 minutes before we're going to log off.
Small doses. Don't overwhelm yourself. I think this was a good step coming onto the live, just getting exposed to some of the terms again. I always say that science is I say it so much I forget it. It's a language first and a concept second. So you have to know the words in order to put them into context, into larger pieces. So the more you can speak the words out loud, hear them, talk about them with other people, it's really kind of removes the stigma of something that might be really foreign to you. Or if you struggle with unit 1 or unit 2, don't ignore it until too late, because bring it back as soon as possible so you can find those holes, reach out to us, and get some help. Awesome. So Tiffany, what can they expect from penguins over the next couple of weeks?
Well, I posted my calendar about 2-3 weeks ago. So next week, actually tomorrow, we start in unit 2. So we will go through unit 2 and hope, I think I have it almost paired up almost perfectly that when we finish a unit, we do a live. So they'll see again my questions showing up in my story. I don't know how many of you are all my followers and are seeing those each day, but there's multiple choice questions and there are short answer questions, as well as at the very end I put a little question box and you can put any questions you have and I answer those each day. Our lives, somebody just asked when our lives are going to be. I worked it out that they're almost every 2 weeks. So every other week we're doing one. There is a span where we're going to do 3 in a row because we had to get around Easter Sunday. I think we got around to Easter Sunday, right?
Yeah, we
were talking about it.
We're on Mother's Day or something.
We're on Mother's Day, but that's okay. That's the day before. That's the Sunday before AP exam if we're going based on the traditional calendar. Because all of our stuff was planned out before they changed the calendar and said, we're going to offer these other dates. So if you go, actually, I think it's saved on both of our streams right now of those dates. But our next one, I believe, is the 1st.
Yep, 2 weeks from today. We'll be back on for Unit 2.
So we'll do Unit 2. And so definitely follow along. There is a review packet that I made that is 245
thick.
pages.
It is thick.
It's thick. But the reason why it's thick is because it breaks it down. So I have gone through the entire CED and I made I can statements. And then I made you questions about those I can statements. Do not ask me. I did not make an answer key for it because it's designed for you as a student to work through it and go through all those questions and make sure you understand, not for me to do the work and you sit there and read it. After that, there is multiple choice questions already aligned to the unit, as well as there's an answer key for that. And then there's free response questions. Don't freak out. Unit 1 doesn't have any free response questions that I could find. Every single thing in there is non-secure. I can't give you secure materials. I like to teach a little too much. And I don't want College Board to pull my accreditation for giving you materials I'm not supposed to give you. So everything is non-secure. So you were able to find all that stuff online if you wanted to, but I have put in a nice, beautiful, thick packet for you if you want it.
We thank you for that. It's amazing.
Yes,
Such so much work.
And if you're looking for it, it's apbiopenguins.weebly.com is where I've posted everything. I have the review from last year. Another teacher, Mr. I believe is a Kramer. I know his first name is Peter. He typed up all my questions last year and put them onto PowerPoints. And so they're on that website. I put my AP Jumpstart that I did with Macro Learning is on there. I've put the live stream that we did last year. RecAP and I did last year. It is a 2-part, and there's a little 5-minute clip in the middle that's missing. You can get on YouTube, but there's like a 5-minute clip. But everything that I have available is on there. Every day I'm exporting my story and I'm popping that up online. It's not on the page yet. I'll probably just put the full unit up there, but anything I have, I'm trying to help you all.
Yeah. So our podcast that come out, biology comes out every Wednesday. And so we have a schedule coming out all the way up to AP exams. We have a full season that came out last year already. So it's the APsolute RecAP Biology Edition, and you can find it on our website, theabsoluterecap.com, as well as all of your podcasting platforms. And all of our podcasts are free, and they all line up to the CED. So they all start with like, we're in this unit, this is our big idea, this is the topic we're focusing on. So you can really narrow down and get that a little closer. They also have accompanying study guides that have answer keys you can find on our website for extra help.
And before we go, I'd like to tell everyone that if you found this helpful, if you thought that either Melanie or I was helpful, be sure to tell your classmates about us, because just because you have a great resource doesn't mean you should keep it to yourself. Go ahead and tell your teacher so that your teacher can tell the students next year and in the other classes, because we're providing free materials for you and we want to help as many students as we possibly can help. So definitely share, let other people know, because we love to help students. Like, we went into teaching to help. Like, we didn't go into teaching for any other reason but to help students learn, because we love to help students, right?
Awesome. Absolutely. No, it's true. And like, you know, we both teach in very different environments. Oh, that's awesome. And we have in different places of the country, and we have people who follow our accounts from all over the world. And we just love to connect with you guys, answer your questions, and point you guys in other resource directions. You know, we mentioned a few earlier, but we have, you know, other AP account friends and other subjects that you might need help with. So although you might come to us for biology, we can also get you to the right place for other subjects too. So yeah, what is the score of the game? I think it's back on. Anybody know the score? Can you pop
Somebody said
28-9.
26-9 Tampa Bay.
All right. Thanks, Kristen.
Okay, well,
we should go.
I think that we have used our hour and everybody wants to get back to the football game, because our halftime hour-long show is over. But it was wonderful to work with you tonight, Melanie, from APsolute RecAP. And we will see you all in 2 weeks.
2 weeks.
Bye, everyone.
Have a great night, guys. Bye.
Bye.